Win at work by communicating effectively with Grammarly.
Meet Todd.
With Grammarly.
I think if everyone is ready, we can probably start now.
Yeah, Ms. Jan, please go ahead.
Yes. Thank you, Singh sir. How are you?
I'm fine, quite fine.
All right. Well, wish everybody a very, very good evening and welcome to our webinar series. I think after the schools have reopened, this is the first webinar that we are having, and with the schools and so on, with their timings, maybe that's clashing a bit, but we'll discuss that in a minute. But welcome to our fourth webinar in our series of 12 webinars for all the teachers who are joining us. And everybody from CSIR-NIScPR, also from Dr. Govind, who is also online. So just to go through a few items and let you know, this webinar is being recorded. For all the students who might be missing it, would request the teachers afterwards when we share the link with you all to pass it on to the students. If they missed it or wish to view it again, it will be available on our YouTube channel. Now, just in order for the purposes of this webinar, the mic, video, and chat have been disabled at this point. There will be a Q&A session, and during that session, we shall open the chat box where you can type your questions. Now, just briefly to let you know about the partners who are coming together for this webinar series. So United Schools Organization of India, which is the organization I represent, we are in registered society and we were established in 1951. And we routinely conduct several activities to advance the social, cultural, and moral standard of schools in India to global levels. All of our activities are national in nature, and they range from extracurricular tests to seminars, webinars now, and programs, competitions, several activities, all of those on a national platform. So we have the honor and the privilege of associating with CSIR-NIScPR. Sir, I won't do justice to the description as much as you would. So if I can request Mr. C.B. Singh, who is the director of the Awareness Division, to speak about CSIR-NIScPR and then about Jigyasa, which will be in the following slide. Sir?
Thank you, Ms. Jan. First of all, good evening to teachers, students, staff of USO, and the staff of Jigyasa in the training division of CSIR-NIScPR. Welcome all. And I wanted to welcome Dr. Govind also. And the different scientists of the CSIR, when they are delivering what the research they have done to the students. So I feel so happy. It's a societal work that we are doing it. So a brief about NIScPR. NIScPR was formed by a merger of two organizations. One is NIScPR and the other is the NIStats. NIScPR, very well known by the three popular science journal, Vigyan Pragati, Science Reporter, and Science Ki Dunia. And NIStats, which is a institute, CSR institute, particularly in the field of the science policy and the research studies. So a platform has been created by the CSIR in collaboration with the Kendriya Vidyalaya Sangathan, and that was coined as the Jigyasa. And this was formed in 2017. And why this was formed? Just to inculcate the scientific temper among the students. We are seeing that the percentage of students adopting or taking the science stream has a bit reduced as compared to the earlier. So we just wanted to showcase to these students that they're the success stories of the scientists who are working to it, to inculcate the scientific temper among them, how inquisitive or what are the daily requirement in which the science has been implied. So all these things has been taken care in the Jigyasa. So Jigyasa was a collaborative project between, or a platform between the CSIR and the Kendriya Vidyalaya Sangathan, but it has been extended to Navodaya Vidyalaya Samiti also and to the state governments also. Now, what does or what happens on this Jigyasa platform? So they're the different models which takes place on the Jigyasa platform, like laboratory visit. Laboratory visit means that the students are coming to the different laboratories. CSIR has around 37 laboratories all over India. So the students are coming and having a visit to the laboratory, what research work is going on, how it is going on. So just for them to satisfy their inquisitiveness that how the scientists are working. The other is the popular science lectures. These are the different models which is prevalent on the Jigyasa platform. And so popular science lecture is just like that we are having each of the scientists or a scientist who are doing the research in the different laboratories and in the different domain. They express or they just tell that what they are doing and how it is beneficial to the country or to the society. So this has been set up using the popular science lectures. Then we have on the Jigyasa platform, we have a summer vacation programs that the students can come to the laboratory for the summer vacation. And sometimes the scientists can go to the school, deliver what is going on in and around in the field of the science. And as well, the teachers and the students can also come to the laboratory. Then we can have we have the teachers workshop. Then we have student residential programs, lab specific activities at the site, like in the laboratory or in the CSIR lab. Certain kind of the laboratory which is safe for the students can be taking place at their respective lab site. So this is all about the Jigyasa. And the future of the Jigyasa is that it has collaborated with MHRD and the IIT Bombay also. And they have come up with a virtual class lab or the virtual lab concept. It's a virtual lab concept wherein the different modules of animation, then the comic stories, simulations can be generated and which can showcase to the students as well. They can access the portal, they can learn it, they can see that how the experiments are taking place. Moreover, if the students are so inquisitive, they can also develop their own content, they can develop their own video or the animation, and that can be uploaded onto the virtual lab. So this is all about the Jigyasa, and this platform, the NISpR, is a platform which is joining the students, which is coming from the USO's help, and CSIR-NISpR is bringing in the scientists, requesting the different scientists from the different labs to come and then to deliver their research work to the students so that they can also have an understanding that what is going on, and they can also develop a desire or a need or to go to or to opt to the science as a field. So this is all about the Jigyasa and about the NISpR. Over to Ms. Jan, please.
Thank you very much, sir. That gives us a great deal of insight, as you know, with respect to what Jigyasa does. Now, for those who are joining us for the first time, just to share with you all what is Science 101. A lot of the teachers and students are already online, and I think they are aware of it, but there might be some new students. So we came up with this concept of Science 101, and we are organizing monthly webinars, and these are going to be started in April and will continue on till March. And this webinar series shall highlight real life applications of science. This is to generate interest in the field of science, as Mr. Singh just mentioned, among students. Very often we study science and we are uncertain of how it is applied in everyday life, but everything has a scientific reason behind it. And this is one of the main reasons why it will help all students to understand the science behind a few things. So just to, as you all know, this is our 4th webinar in our series. And the title of this webinar is Drug Discovery and Safety Studies. So going through the session of today's webinar, we've changed the order a little bit. We are first going to watch a quick video. After that, what we will do is I will also take on the honor of introducing Dr. Govind Yadav, and he will then speak about various things. But before we delve into that, just a quick video so you all sort of get to know what it is that we shall be doing. So bear with me 1 minute.
Ms. Chan, if any issues coming in the streaming the video, then that can be taken up later, and we can start with the.
Is it not streaming, sir, the video?
Actually, I'm not able to see it. I hope others may. Suman, may please comment on this?
No, sir, it is not streaming.
No, it is not streaming. Actually, we are not able to see it. Only the poster is coming.
Just 1 moment.
Yeah.
How about now?
Not yet.
Okay, just 1 moment.
Yeah.
Just 1 moment. I think we'll by then, if I could perhaps introduce Dr. Govind until that time. Apologies for that. We think we have a hang of technology, but it always gets the better of us.
Yeah, please.
Sorry about that. So we'll play the video in a minute. Until then, just like to welcome Dr. Govind Yadav and thank him profusely for consenting to host this webinar series in Science 101. What we say about Dr. Govind is very less compared to all his magnificent achievements. Dr. Govind is currently the principal scientist and associate professor at the Academy of Scientific and Innovative Research. This is the Pharmacology Division at CSIR-IIIM, which is the Indian Institute of Integrative Medicine. He has more than 18 years of experience in this field, and he has several publications to his credit. He's also co-authored several chapters of several books which range in various topics. Those topics are his main areas of interest, which are preclinical drug discovery and development, natural products, animal models, immunomodulation, mutagenicity, carcinogenicity, infection and immunity, mammary gland biology. These are a few of his areas of interest, and I think it takes a lifetime to have all of these interests and to find the time to publish and co-author all of these books. So really, we are very honored to have you, Dr. Govind, amongst us today. Thank you so much for taking the time to impart this knowledge, which you will be sharing with the students and the teachers. We are truly honored to have you here. And perhaps we can start with your presentation, sir, since we seem to be having certain video challenges, and I don't want to hold everybody. Dr. Govind?
Yes, please.
I think the video is on.
You start from the beginning.
Yes, sir. I think we're trying to do that. Just 1 minute.
Yes, the video has been available and it is being visible. Right.
Yes, yes.
CSIR, Indian Institute of Integrative Medicine, IIIM, Council of Scientific Industrial Research. CSIR is India's primary national research and development organization, IIIM, comprising of Council of Scientific Industrial Research, CSIR, 39 India's primary national research and development organization, by comprising of 38 state-of-the-art national laboratories, personnel, 39 CSIR's role in escalating scientific innovations by 4600 scientists are the crowning features, 8,000 technical the nation is fully CSIR's role in escalating scientific innovations is extremely fortunate to have the crowning features, the greatest natural wonder of Earth, fully as it provides us with exceptional flora and fauna. India is extremely fortunate to have Himalayas, the greatest natural wonder of Earth, as it provides us with exceptional flora and fauna. Indian Institute of Integrated Medicine, supported by 46 scientists of CSIR and 8,000 technical research on drug discovery. Institute of Integrating Scientific Innovations is Medicinal Institute of CSIR with prime focus of research on drug discovery from natural products including medicinal plants and microbial species, and enabled by biotechnology to develop anti-inflammatory drugs, anti-products of high value from Boswellia serrata. International resin of Boswellia species is used as anti-inflammatory and cultural ceremonies drug and from Boswellia serrata. Time immemorial. Resin of Boswellia species is used as incense in religious and cultural ceremonies, and in medicine it is also widely used in leading detergents and cosmetic industries. The extract of plant can produce anti-inflammatory drugs, anti-products of high value from Boswellia serrata. International resin of Boswellia species is used as anti-inflammatory and cultural ceremonies, drug and from Boswellia serrata. Time immemorial. Resin of Boswellia species is used as incense in religious and cultural.
Having some little bit issue in video and the sound. Perhaps, Dr. Govind, may I request you to start with the presentation first?
Okay.
Sorry about that. By then, I hope we'll fix everything and we'll play this video before we conclude.
CSIR, Indian Institute of Integrative Medicine, IIIM, Council of Scientific Industrial Research, CSIR, is India's primary national research and development organization, comprising of 38 state-of-the-art national laboratories, 39 outreach centers, 5 units supported by 4,600 scientists, and 8,000 technical personnel. CSIR's role in escalating scientific innovations and technologies are the crowning features for which the nation is fully indebted. India is extremely fortunate to have Himalayas, the greatest natural wonder of Earth, as it provides us with exceptional flora and fauna, providing abundance of life-supporting herbs, species, oils and minerals. IIIM, Indian Institute of Integrative Medicine, Jammu, is a premier institute of CSIR with prime focus of research on drug discovery from natural products including medicinal plants and microbial species, and enabled by biotechnology to develop technologies, drugs and products of high value for national and international markets. Plant-based anti-inflammatory, anti-arthritic drug from Boswellia serrata. The resin of Boswellia species is used as incense in religious and cultural ceremonies and in medicine since time immemorial. It is a moderate to large-sized branching tree that grows in dry, mountainous regions in India. It is also widely used in leading detergents and cosmetic industries. The extract of plant can reduce pain considerably and improve knee joint function.
Yes.
Microbial fermentation process for the production of DHA, docosahexaenoic acid, is traditionally obtained from fish oil, and it is an effective medication that slows blood clotting. Therefore, it is also known as anti-platelet drugs. It is a great source of omega-3 also. The fermentation technology of CSIR-IIIM is the latest, and the facilities from lab scale to pilot production are well organized. CSIR-IIIM is involved in various consultancy projects and is helping in technology transfer and training to various companies and institutions. Development of a new formulation, Reserine, with enhanced anti-tubercular activity. Reserine is the first anti-TB drug researched and developed in India. Reserine is the outcome of path-breaking research carried out jointly by CSIR Jammu and Cadila Pharmaceuticals Ahmedabad. It was developed with the objective of overcoming the limitation of conventional rifampicin. More than 90% patients development of a new formulation TB with lesser side effects, better safety profile and low cost. Live One, new hepatoprotective drug developed.
This is my little contribution. Maybe a student will enjoy this one. So thank you USO and NISPR for giving me opportunity. Okay. So that before I go to topic, just little bit. What is the scientific research and what is the present scope? Career options. So students can see what is the career scope in scientific research. So after graduation and after masters, after they start research from sometime in master research, but in PhD the full-fledged research, and after that they can go to industry to do their research. They can become a professor in university, lecture in a school, lecture in colleges, or scientists in different organizations like agriculture institutions. They can be a CSIR scientist. They will take opportunity to do wonderful science. Maybe they go to space research. And scientists in any scientific organization, other than this, what we discussed here, they can start their own startup using their scientific experiences, and they can become a patent officer, law officers, and other. So I would like to, this is the CSIR already in video. You can see there are 39 laboratories are there. There are outreach programs are there. Innovation, 38 national laboratories, 39 outreach centers, innovation complexes and units. More than 4,600 scientists are active scientists are there. So this is other than this, the pan-India distribution of the research laboratories of CSIR. Indian Institute of Integrative Medicine, located in Jammu, is also one of the part of the CSIR. So the CSIR Indian Institute of Integrative Medicine, this institute was started by the great Indian pharmacologist Sir Colonel Ramnath Chopra. He is the father of Indian pharmacology, and his contribution is more actually. He started drug research laboratory in 1941 before the independence. Later on, it merged with 1957. The CSIR took over this, and he was the first director of this institute. And he worked on the Rolfia serpentina, and later on, from Rolfia serpentina, there was a reserpine, anti-hypertensive drugs. So he is the great man. So the topic is drug discovery and safety. Everybody knows about the drug discovery. Scientists are engaged to discover new drugs. They synthesize, they can isolate from the natural products, but safety is very important. If there is no, a drug is very effective, but there is no safe, then this will be no use. So I gave two examples here. One is ivermectin, the structure you can see here, and another one is the thalidomide. Everybody knows thalidomide incidence in 1950 and 1960. So there are the two important aspects, the efficacy and safety. So based on this, the drug discovery and the safety is also very important along with the drug discovery. So what is the path of the drug discovery? Scientists in everyday life, he think and they get an idea or plan of the project. So he discussed with his team about his idea, or other members can also suggest what is the new idea. So after that, they will implement it in laboratory. So they can synthesize the drug, they can isolate from the natural product, they can isolate from the bacteria, they can isolate from the marine sources. After that, there will be some bioassays. Bioassays include the in vitro studies in laboratory and studies on the animals for efficacy and safety. So what is their plan? They can execute it. So they will first check what is the target of the drug. Okay, so any disease they will take, and then what is the target of that disease? So they can hit the target. If target your drug is showing effective in the inhibition of that target, responsible for the disease, then the compound will move to the in vivo study. In vivo studies means the studies on the animals. So any animals, you will check that the activity of that compound is promising or not. It's effective or safe. Safety is there or not. So after that, these are the data in vitro and in vivo, called the non-clinical safety. After that is safety study. Safety is a number of the tests which has formulated by the OECDs, suggested by OECDs and the FDA, what they want from the researcher. So those non-clinical safety data need to be generated. So after these information, non-clinical safety data, so the one draft will be formed for investigational new drug, IND, investigational new drug, or first in human. The compound having the what data in this draft. So there will be the pharmacokinetics data, the potential effectiveness data, what is the concentration of the efficacy, what is the dose, what is the safety, what is the tolerability of margin of that the compound or drug. After that, if there is everything is okay, the drug, proposed drug is safe and effective, then it will go to the phase 1 clinical trial. Phase 1 clinical trial means this will be in human. The non-clinical safety is on the animals, and after that clinical trial will start on human beings. Little bit after the permission of the regulators. So phase 1 clinical trial is the compound or drug will be first will be tested in 20 to 100 people. After that, it will be tested on the 100 to 500 people in phase 2 clinical trial. If all are okay, then the compound or drug will enter in the phase 3 clinical trial. So all these are the early development. So by using this data, again, the scientist or the company which formulating this drug, then they can approach to the regulators to say their data. After this data, so the company or any scientist who are working on this drug, developing this drug, they can submit the new drug application. What is NDA? New drug application, or market authorization application. So what this include all the data of the safety and efficacy, any the labeling of the drug, then all the controls, maintenance, drug quality, drug identity, and the strength, quality, and purity of this data. So all data, safety and efficacy and other labeling and other data, which that is list of the regulators in that format. The scientists who submit or any company who are formulating this drug, they can submit the data to the FDA. Okay, that is the regulator. Then compound will enter in the phase 4 clinical trial. So after that, it will be registered in the different regulatory agencies. And like in India, Drug Controller General of India, DCGI, that we give permission. So after that, the compound or drug, the new drug can be registered in the different regulatory agencies, and then it can be marketed as a new drug. So this is the process of that. So here, the more clarity is here. The basic research actually, you can say in drug development, discovery and development, the 10 to 15 years required from basic research to the drug float in market. So you available for the patients. So 10 to 15 years it's required actually. So in basic research, different the disease, what is the pathogenesis of disease according to the pathogenesis. So what is the protein or pathway, all those responsible for the actually disease progression. So using the hundreds of the compounds, thousands of the compounds, the people test for the compound or is effective to inhibit the responsible target for the disease. So After that, this after basic research, several years of basic research, different bioassays setup, standardization, all those. After that, the compound will enter in the drug discovery research. So, like for example, like the candidate drug having 5,000 or 10,000 molecules, after in preclinical testing, so only 250 will remain. Means that there is a large number of the failure. After that, the phase 1 clinical trial is start. Only 5 to 15 out of 10,000, 5 to 15 molecules will be entering the phase 1 clinical trials. So, like this, the phase 1 clinical trials we earlier discussed, the 2,200 people will required, and the phase 2 clinical trial number of volunteers will be the 100 to 500. And in phase 3 clinical trial, thousands of the people required. The drug will be tested on for efficacy, safety, and all those data. So FDA will review all the data for safety, efficacy, labeling, the tolerability doses, what is the margin of the safety, all those things. Then after that, FDA will approve. And compound will enter into the market, drug enter into market, and this is called the phase 4 clinical trial. So how drug is start actually? So there are three things we need minimum. So one is the compound. Compound means so which will work, which is the main ingredient in the drug. So drug is the formulation actually. In formulation, one is the active principal ingredient. So active principal, and then others are the to make it drug. So one need the compound. So compound come from the different sources. Maybe it can be synthesized like this. First, synthetic chemicals can be synthesized in the chemistry lab. To the scientists who working on total synthesis work, they can synthesize the compounds which will work on different diseases or the target of the disease. Another source is the medicinal plants, is a larger source. So in India, China, and other countries, they are having rich source of the medicinal plants. We are having traditional knowledge in Ayurveda, different the local people, like in villages, they are having good knowledge, the traditional knowledge of which plant will work in which disease. So based on this, the information, there is a traditional knowledge, the information available in literatures or maybe in the experienced practitioners of in villages and of traditional medicines. So there are different plants having rich source of the different the molecules. So we are looking for we are extract from the different bioactive compounds, which further will be tested for different diseases. So that work or not, that effectively inhibit the drug target or not. So this is the natural product to come from the medicinal plants like Himalaya and other. There are thousands of medicinal plants in India and other countries. So another area is microbes. So different microbes also producing the different compounds, drugs. Earlier we seen different antibiotics comes from different bacteria. So one example is like Streptomyces griseus. This is the colony. From this colony, this is the compound streptomycin isolated. So everybody knows that this is the good antibiotic and like in first, this is the compounds which work on the gefitinib, which is gefitinib, and gefitinib is active against the small cell lung cancer. The target is this is the kinase inhibitor. So we need a compound drug. We need the drug target, means the particular disease having the particular target which need to be inhibited. So you can stop the disease or you can control the disease. So they are the compound, the source of the compound, Then we can isolate the compounds. So we need the pure compound. Then after that, we need a disease target. So that we can then this compound will interact with that drug target. So this is the process of the how we can isolate the compounds from different plant sources. So we know the sources. They are from traditional knowledge. There are different sources. Like one of them is the medicinal plant. So medicinal plant, how we can get from the medicinal plant drug from medicinal. So first, like we are having the plant material, then we can dry it. First we clean it, then we dry it. Then different methods of the conventional methods. So we can extract by chemically or by different other methods. Different solvents we can use, and the non-conventional methods are the like different techniques are available. So by using these techniques, we can extract the different make an extraction from the different medicinal plant. So after that, we can use because this extract contain the different the solvents. So we have to evaporate those solvents. So this is the rotavapor. So we can evaporate those ones. After that, we can get the final extract. So final extract we can use for the purification. Okay. So each extract, there are two methods here. We can either directly use the extract for different diseases to test if it is effective in different diseases or not, or we can go for purification. So after purification, we can after that each extract will be again processed. We call it the bioactive guided fractionation. So after purification, so after that we use the different equipments like HPLC, LCMS, NMR to identify what is the structure of the disease and how much concentration is available. So each peak represents the one compound. Here the same thing is here. So little bit detail is there, like plant material extraction. Then this is the bioact. So first we do the activity with the extract, and then again we will process the extract. So like in first extraction by different methodologies, then we do the bioassays like antibacterial activity, antifungal activity, anti-cancer activity, anti-inflammatory activity, different enzymatic assays. When we found that okay, this fraction or this extract, there's a part of the extraction is the active, then we can test only that extract. Further we can process it. So sample will be prepared. Then activity on oriented separation of the compound we start. Then after that, the activity oriented separation, the structure will be elucidated. So we use the different methodology or different equipment for this, like UV diode, MS, NMR, HPLC, MSP, GCMS, HPLC, and other. There are a lot of things. So after that, we found a single molecule. This single molecule is having a one structure. Okay. So then after that, like this, these are the plant material, different plants. These are the source of the drug. And then after that whole process, after the whole using the whole process of the extraction, we get the single compounds. Like each having thousands of the hundreds of the compounds, but what we are looking for, which is compound is the higher concentration in the extract and which is active. So we can get from the bioactive extracts. Similarly, like from plants, this is the bacteria. So similarly, we can also extract from the bacterial colonies. We can make a bacterial colony. We can expand in the liters of the culture. Then we can optimize the conditions, growth conditions like temperature, like RPM, different medias, which media support this. So like this, we can first cultivate the particular strains of the bacteria, and we can see the activity directly from the extract, and then we can find that similar in plants. We can also find the structure of the bacteria. Then we do the bioactive, the bioassays. Bioassays are similar, like antibacterial activity, cancer activities, and other assays. So after that, purify the compounds with different equipments. So we can, after that, we can elucidate the structure of the compound by using the like NMR, X-ray, all those things. So like you can see here, there are different antibiotics that come from the different organisms. Like penicillin come from the Penicillium species, streptomycin from Streptomycin gracius. So like gentamicin, some micromonospora species, the rifampicin from Streptomyces species. So like this is active against the different gram-positive, gram-negative bacteria, different tuberculosis, fungi, they are active against this. So each compound or each antibiotic having different mode of actions. Means how it will work. So some work will like if you see the structure of the bacteria, so there is wall is there is cell membrane is there, the proteins is there, different nucleic acids are there. So how these compounds act against so we can classify these are the antibiotics. So like this, you can see the bacterial cell here. They are DNA, mRNA is there, ribosome is there. So different the targets are there for these antibiotics. So there are a group of antibiotics which work on the cell wall synthesis. Then they stop the cell wall synthesis. They interfere in the cell wall synthesis. So bacteria will come. Some work on the DNA. Some work on the RNA. Some on the DNA directed RNA polymerase, like rifampicin, streptomycins, like protein synthesis. Some are working on the protein synthesis, like they stop the protein synthesis, like erythromycin, chloramphenicol, clindamycin, lincomycin, like this. Some are also inhibit the protein synthesis, like that. There is list of the compounds. So different antibiotics or different compounds having different targets. They act against different targets of the disease. So this is one example. Ivermectin. This is a wonderful drug. Ivermectin is the wonderful drug. It's come from where is come from? The Japanese scientist in 1981. That Dr. Satoshi Omura, he isolated this antibiotics called this from bacteria actually. So bacteria come from the soil. So in Tokyo there is the one golf course is there. So near that golf course is the soil of the golf course. So he used this golf source, take in the laboratory and in media, and then grow this type of colony from bacterial colony. Or after that, they isolated this type of the structure of the compounds. And first, this ivermectin was used with the help of the Merck laboratory for the parasites, like different ectoparasites, different endoparasites. You know the parasitic diseases responsible for scabies, like endoparasites. So that work against that in animals. After that, it was identified that this ivermectin work against different bacteria also. Okay. So not only it was tested in the animals, but after that this was recommended for the human being also. So due to this wonderful discovery, the Dr. Satoshi Omura from Kitasato Institute of the Japan, he got Nobel Prize in 2015 in physiology and the medicine. Along with the Japanese scientist, the William Campbell from the Merck laboratory, he also got Nobel Prize with him because he worked with him. So this ivermectin was approved by the FDA as an antiparasitic in 1981, and there was one disease called the river blindness. It's also called in human being in Northern America. So there are thousands of the people every day suffer from this river blindness. So 1987, it was again approved for the river blindness. So these two discoveries, antiparasitic and river blindness, so Nobel Prize Committee recommended in 2015 for Nobel Prize for this discovery. So here, like we learn about how we can have a compound or which is effective for different targets of the diseases. Maybe it will from synthesis, it may be from the natural product, maybe it is from the microbial world. So after that, we need the compounds and compound tested in vitro in laboratory for different disease targets. Okay. So for after this, we need the animal study. Means the animal is the before we jump to human being, we can not directly test our drug, the potential drug which is effective in vitro studies in laboratory. So we first test in the animals. Okay. For what we will test? We will test for effectiveness of disease. This is working against the disease, means the inhibiting the disease. After that, we will test its safety. Drug is safe or not. So it includes the this is ADME, absorption, distribution, metabolism, and excretion, pharmacokinetic study, then toxicity studies in animals. So animal should not show any the death or any side effects like weakness, reducing body weight, or any change in the blood parameters, maybe change in the biochemistry parameters. Or if there is any death on this, any short of the reduction in the body weight during the experiment, so we can test for histopathological examination. So what is the effect of that the drug on tissues? Okay, so toxicity studies will be so safety and toxicity, different organ systems we will check for this one. What this slide is showing, this is for one animal model which I work on. So this model for the cancer. So generally, what is the tradition is the xenograft. Maybe you heard that we use the immunocompromised mice. There is no immune system. T cells and B cells are there. So immunocompromised mice we check, and then we try to grow. We inject the human cancer cells in mice, so there is tumor will be developed. And so somebody asking that which topic you are talking. So this is the drug discovery and the safety studies. So what is the different drug stages of drug discovery? So we first isolate the compounds, then we find out the different the drug target of different diseases, and then after that we will use the in vitro studies, then in vivo studies. So in vitro studies, we use the different cells, different ELISA, different bioassays to check that the compound is effective against the particular disease or not, particular target or not. After that, similarly, we are having the animal model of biological system to check the effectiveness and the safety of the drug. So here in this particular model, like this is for cancer. So we can, this is the, you can see the red dots which represent the cells, cancer cells. We can fill in this fiber. This is the hollow fiber, 0.2 micron diameter. So we can then transplant in the immunocompromised mice. After that, we can supply the drug of his interest. So what is the difference of this mice, this hollow fiber mouse model for drug screening? So we can reduce the earlier, if we compare with the genograft assay. So this system is only 10 days assay, or 3 cell lines we can use in 1 animals. So amount of the compound required for the genograft study, we can reduce here. So we can use the 2 physiological compartments. So we can check the compounds effective in subcutaneous and intraperitoneal compartment also. So like this. So like this, different animal models are here. Like you can see the number of cancer cells, like colon cancer, colon 205 and SW620. These are the colon cells. Like for breast cancer, we use MD, MA, MB231, MDA, MB431. There are different cancer signs for lungs, for ovarian cancer, for CNS, melanoma. There are the group of cells selected for the screening of the compounds. So other animal models, like this is the wound healing models. We can test the different compounds, different extract from the natural product or from the microbes. We can test for the wound healing. The drug is effective in wound healing or not. The reducing the days of wound closure compared to control and without drug. Okay, this is the mouse model for the mammary gland diseases. So we induced by lipopolysaccharide, which is inflammatory. So then we test our drug here. So you can see here the inflammatory cells, the infiltration is reduced. The inflammation is also reduced. This is the sickle cell anemia. This is the mice. This is the declay mice. So I procured from the USA. To test that sickle cell anemia. Now other group is working on it. So this is the slides of the histopathological slides. You can see the changes. The spleen is large and other organs is also having. So earlier we already discussed that to start the drug discovery, first we need the target identification and validation. Then number of compounds we can test either from the synthesis, from natural products, and there, or we can procure from the different chemical libraries. Okay. Then after that, so identification of the hits, then the assay development and screening, hit to lead, and lead to the lead will be optimized, and the in vivo and the in vitro assay will be conducted. And then after that, after in vivo studies and safety and all those parameters, the compound entering the clinical studies. So like this, what is the time required? What is the time required for each cycle? And then what is the probability of the success of different phases? We already discussed this thing. So how we can decide that the compound is safe or not? The safety. What is the safety? So the first class of the compound is the good efficacy and the toxicities will be low. So this is the good class of the compound to take this compound or this drug to forward to become a drug. The compound become a drug required the good superior efficacy against any disease, which can able to inhibit the disease, progression of the disease, and the compound toxicity on the human being or animal should be low. So this is the superior compound to take it forward for drug development. So another second class of the compounds, efficacy is adequate, but toxicity high. So we cannot take this compound for drug development. Another third group of compounds, efficacy is adequate, toxicity is manageable. Okay, so we can work on this modification of these compounds so to increase the efficacy and reduce the toxicity. But some compound like class 4 compounds, they are the low efficacy against any disease, means they cannot able to means only little they can target any disease or reduce or inhibit the drug or progression of the disease. And there are high toxicity. So we cannot recommend these class 4 compounds for the further drug development. So to reduce the time and the animals, nowadays the high throughput studies will be in talk actually. So in laboratory we can do like this, like tissue buffer chamber for the different to check the action of the drug on different tissues. Okay, so one chamber having the plasma with drug, and another is the chamber is the homogenized tissues. So we can check the action of these compounds on different tissues. So in between these two chambers, there is semipermeable membrane, so compound can pass. So the second B, the B figure is showing the different organs on chip. This is the new technologies coming. So so we can reduce the number of animals used for the different studies. So these like we can check the efficacy or safety of the our compounds on lung, heart, kidney, bones, liver, gut. So all these organs will be on chip. So we can directly test the safety of drug on chip. So this is the new methodologies, not reached in everywhere, but the scientists are trying to validate this. So these things we already discussed. So the target identification and validation, first step, high throughput screening, hit identification and discovery, assay development and screening. We need the good assay, effective for to check the effectiveness of the disease. So there should be effective target. So hit to lead, then lead generation and optimization, in vivo and in vitro assays, clinical process phases and regulatory approvals. So they are here that we discussed in first figure, different phases of the clinical trial. What is their role? Phase 1 clinical trial, 20 to 100 healthy volunteers are required. So purpose of this is to see the safety and doses of the drug. The length of this study is several months. Approximately 70% of the drug move to the next phases in second phase. So in second phase, we need to screen the compound on to check the compound's efficacy and safety on 100 to 500 patients. So here also efficacy and side effects we have to check. And the third clinical trials, we need thousands of the human being or volunteers to check the efficacy and safety of the drug. So only 25% of the drug can go to in the next safety phase 4. So what is other parameters required? Like pharmacokinetics and pharmacodynamics data, bioanalytic method validations. They are the part of the drug development process. Regulatory approval and post approvals, FDA regulatory approvals we have to take. So different These are the list of the different tests we have to test for the safety, like systematic toxicity studies, male fertility testing, female reproduction, development, teratogenicity, local toxicity studies like dermal toxicity study, photoallergy, vaginal toxicity, rectal tolerance, ocular toxicity studies, inhalation toxicity studies, allergenicity, hypersensitivity, guinea pig maximization test, local lymph node assay, like in genotoxicity. So the compound what drug we are taking should not be genotoxic. So we have to check the drug with different assays like chromosomal aberration assay, micronuclei assay, comet assay, sperm abnormality assay, test for the gene mutation in bacteria, like we call the popularly known as the EMS test. So we can check the compound or drug, which can be drug, which is effective, but is it safe or not? Is the toxic safe or not? So we have to check for different mutagenicity assays, safety pharmacology system, the organ system. So we have to check the cardiovascular system, the blood pressure, heart rate, electrocardiogram, the QT interval, Hurg assay. And similarly, we have to check the safety of the drug for the central nervous system, like motor activity, behavioral changes, coordination, sensory and motor reflex responses, body temperature. Similarly, we can also test the drug or compound in the respiratory system. So compound should be or drug should be the safe to the respiratory system also. So every system should be safe actually. So this is the purpose of the drug safety actually. So respiratory rates, the compound is affecting respiratory rate or not, hemoglobin, oxygen, saturation, tidal volume. So these are the requirements for the IND submission of any new drug. So we talk about the thalidomide. In first slide I mentioned this. This is the real example of the drug safety actually. So why drug safety is required? So once in drug discovery phases, the scientists conducted the efficacy studies and the normal safety studies, but he didn't include the animals for the pregnant females. So that's why once the pregnant female take this thalidomide for the morning sickness, nausea. So after consuming this thalidomide, that the different deformities observed in the newborn childs. So this was the major blunder mistake by the scientists. So he didn't include this assay during the safety studies. So why safety study is important for the drug? So you can see this is it's a great example to understand actually. So this photograph, this is the United States President Kennedy. So he's giving the presidential award to in 1962 FDA pharmacologist, the Francis Oldham Kelsey, or blocking cells of the thalidomide in the United States. So she played the major role to stop the sale of the thalidomide, and after that the thalidomide withdrew from the market actually. So why safety is important for any drug? So these are the number of drug. Although there are the provisions to check the drug is safe or not, there are safety data. FDA wants safety data along with the efficacy data. So there are the number of drugs you can see here from the 1950 to 2011. These are the number of drugs which are withdrawn from the market after that actually. So here also I'm just accumulated here in the list out the significant withdrawal from like in just 20 years, last 20 years, the number of drugs due to the safety concern. So that's why the safety is very important along with the efficacy of the drug. So we will take few examples from these safety studies. What is the market drug withdrawal from the market? So we will take some examples. Like recently in 2020, this is the gel actually come from this euphorbia, the peplus plant. This plant is found in Europe, Northern Africa, Asia, Western Asia. It is typically grown in the westlands. Like this is the rock in the rock bottom of the rock is growing actually. So from this plant, the scientists did laborious work and isolated this compound. The called is ingenol mebutate, sold under the brand name of the Picato. So and this is the actually the marketed product Picato. So this is used to actinic keratosis. This is the disease where the skin is the growing uncontrolled. The keratosis. We call it. This is the name of the keratosis. This disease, this condition. So the people applying on this the face, the back, and the extremities to get relief. But there are several reports that after using this Picato gel containing the agenol mebutate, so there is the this is skin cancer inducing properties. So temporarily it was a withdrawal from the market in January 2020 after 20. So again again it will be tested. Again the safety parameters will be submitted, and then after that, if everything will be okay, then this compound again will be this drug again will be permitted by the FDA. So this is the real examples of the safety actually, how we can after the long list of the safety studies required for the submission of the data to the before we get the approval from the FDA. So another one is the recall of the recently actually in 2020. So this is the Genentech actually. Why Genentech is recalled actually? They are showing some side effects. So other than this, the Genentech people use the Genentech for the acidity and other problems. So there is a recommendation that the NDMA is the compound which is carcinogenic actually. So in once some drugs having this compound in less concentration, but during the storage, the concentration of the NDMA increase. So nitrosodimethylamine is NDMA. So increased concentration of the NDMA are the carcinogenic. That's why this 2020 this medicine was recalled from the market. So this is also one example of the drug safety actually. So need to be checked. Similarly, lorcaserin, the marketed as a trade name of Belviq or Belviq XR. To reduce the calorie or weight loss. Okay, this was permitted by FDA in 2012 to reduce weight loss reducing medicine. But after that, there are complaints against this drug, that is it's having uncertain increase the risk of cancer. So due to the risk of cancer, this lorcaserin containing Belviq was withdrawn from the market actually. So you can see here the many drugs withdrawn from the market due to the safety issues Some are I also important drug also mentioned here having the heart problems. So once people take it, the heart problem is started actually. So valvular heart disease, torsade de pointis, this is the ventricular fibrillation, liver failure, and other carcinogenic also. So different drugs they're recommended by like for the hepatitis suppressant, antihistaminic drugs, cardiovascular drugs, antihistaminic, diabetic, gastrointestinal drugs. So they are having some safety issues. That's why they withdraw from the market actually. So here you can say some drugs withdrawal due to the this safety, this complaint that the torsade de pointis, this is the ventricular fibrillation actually. What is the reason for ventricular fibrillation? I'm trying to explain here. The for the cardiac safety, the ICH guideline S7B. So the HERG channel liability in preclinical development. So they are the different the cardiac ion channels. So one of the cardiac ion channel is the HERG. Okay, so what is the function of this? This HERG delay the rectifier potassium current, IKR. So some drugs which inhibit the IKR potassium current, or due to this inhibition, there is block cardiac HERG potassium channel. So that's why if you see here, there is one ECG graph is there. The normal ECG is like this. So there are 4 points, the P, Q, R, S, and T. So there is a time period of the each wave. So how much time the P will be there, then reached up to the Q, and the Q to R, and R to again down to S, and again reach to T. So there is the curve there. So once it is a QT interval, again this cycle repeat in the same way, PQ, RST, PQ, RST, PQ, RST. So if Q, this QT interval, Q to T, Q, R, S, T, this will increase, then this is not safe for the heart actually. So this is called as long QT syndrome, the disorder of ventricular repolarization. So this is the predisposing effect that individual to cardiac arrhythmias. So in one term, we call it the ventricular fibrillation, and there is sudden death due to this long QT syndrome. So some drugs are like here, you can see the different drugs which are earlier in market was responsible for this QT syndrome. So like the two very good drugs for different intestinal disturbances was approved by the FDA. We call like the cisapride, propulsid, and tefendalide. And then the Selden, the marketed and the trade name of the Selden and propulsid. So you can see here, this is the red wave is the T wave. This is the extended actually, time period is extended actually. So its limit should be limit here. Then, but due to the prolonged QT interval, so this T wave is the extended actually. So this is the region of the long QT syndrome and responsible for ventricular fibrillation and the death of the person who was taking the cisapride. So this is overall. Thank you very much for having a patience and listen my presentation. Yeah. So over to Dr. Nina.
Thank you so much, Dr. Govind. I feel very flattered every time we have one of our webinars, and the main reason for that is because I get called doctor a lot. Not yet a doctor, but I must say that it feels very good to be called one. This is a huge push for me to also pursue a PhD. But thank you very much, Dr. Govind. We often, I believe we often do not realize how much of science is all around us.
Yes, please.
And how much of science is a part of our everyday lives. So it's great to get a perspective like this when we know how the medicines affect us and so on and so forth, as you've just said, in case of a heart patient. So with that, thank you so much, Dr. Govind.
Safety is very important.
Yes. So perhaps
you can inform your schools, and they can write me on my email if there is any questions, and after that also after that this webinar they can interact with me.
That would be great, sir. But maybe if we have time for a couple of questions, if the teachers have, or if the students can ask a few questions, perhaps we can open the chat box. Ms. Jolly, can you kindly open the chat box?
Yes, it's open, ma'am. Open.
Ms. Jolly, I think it's still turned off. If we can just have it turned on again.
It's now open. Oh, 1 second. Yeah. Done, ma'am.
So we have a question from Ms. Neha Kaushal, and I must say it's a very interesting one. Why every medicine is tested on mice, sir?
What is the name of
Miss Neha Kaushal?
Yeah, Miss Neha. So Miss Neha, animals are playing their role actually. They don't want you to suffer actually. So they are suffering. So like this is this was the talk on drug safety. So drugs should be safe. How we will test? So either we can on cellular system we can check on in laboratory, then after that we can check in the biological system, where is all physiology, all organs are there. Like in like human, in animals, there are all organ system is there. There is heart is there, intestine is there, the lung is there, liver is there, where drug is metabolized. So if any side effect and toxicity is there, so we can stop that drug there at that stage, not reach to you as a patient. So this is why mice is required actually. So in one slide, we displayed here what is the new development, the organ-on-chip. So organ-on-chip, it means so we can test what we are testing in mice before we can reduce. There is no alternative of the biological system, but we can safety studies or toxicity on organ system we can check on the organ-on-chip. So this is the alternative actually. Yeah, I think, yeah, over to you.
Yes, I guess that answers the question. She says, thank you, sir.
Okay.
Any other question that anybody has? So while they are asking for questions, if I may ask, this is a very interesting area of science, which is pharmacology. How much of chemistry and how much of biology? I think these are the two main subjects if students want to pursue this field. These are the two primary subjects that are required.
Yeah. Actually, the chemistry means you have to what is drug actually? Drug having the one is active, the principal ingredient. Like you are taking paracetamol for reducing the temperature when you have fever. So where paracetamol will come from? It is from either synthesis. Okay. So chemist will synthesize, not biologist. So that's why chemistry is required. After that, biologist will test in the large number of assays, biological assays. So both are important. where a student enjoy, they can choose according. Or Jigyasa having giving opportunities to visit the labs actually, so they can interact with the scientists and see the lab. So accordingly, they can, according to their interest, they can decide. Yes, over to you.
Right. Thank you, sir. So if there are no other questions from the teachers or the students, so if you permit me, everyone who is online, we wanted to check, I believe the timing of the webinar at 4 p.m. We have heard back from schools to say that it is a little early because students do not get back home on time now that all the schools have reopened. So we have got suggestion from some schools requesting us to move the webinar to 5 p.m. For those of you who are online, if you can tell us really quickly in the chat box if 5 p.m. is better, because we have heard that students don't get back home until 4:30. If there's anybody else who can think of this later on and can let us know via WhatsApp also, that would be great. We can change hopefully the timing from next time onwards. So please do let us know if 5 p.m. is a better time. And in terms of next steps, our next webinar will be scheduled for Friday, which is the 27th of August. As usual, we will share the details with all of you later on. As is the practice, the Microsoft Teams meeting link will be shared with for the teachers and the YouTube live link for the students to join us. So if there's no other question, we'll conclude for this evening. So thank you very, very much, Dr. Govind. Thank you for your time and for sharing with us, giving us a sneak peek to everything that happens in the world. Very a great overview of what happens and how the medicines are formulated, how safety precautions and so on are taken. So thank you very much for that little sneak peek. I do apologize that we were not able to play the video because of some issues. We shall circulate the link to the video in various chats as well. I'm going to paste it and put it up on the chat right now. Students can view it, and we will also share it via WhatsApp so teachers can circulate it with their students. Singh sir, Dr. Suman, thank you very, very much for helping us organize this. I don't know if you want to say a few words. Dr. Suman, are you online?
Yes, can you hear me?
Yes, we can, Dr. Suman.
Yeah, thank you. And I would like to really thank Dr. Govind for immediately accepting this, and it was really very, very informative. And I do hope, yes, for the students and as well as the teachers, it must be very, informative and useful also. And he has also given the opportunity to interact in future, so they can grab this opportunity to visit his lab also and to interact directly with him. Yeah, thank you. Thank you.
Great, thank you. Singh sir, are you online? Right. All right. I think if there's nothing else, teachers and students, if you have any questions, please do send it to us via email or via WhatsApp, and we will get that answered from Dr. Govind. Dr. Govind, thank you very, very much once again for being part of the webinar, and hopefully we'll see you at some point at one of the CSIR labs or on another webinar or something around those lines. But thank you for taking the time. Thank you so very much.
Thank you very much.
Have a good evening, everybody. Yes. Have a great evening to all of you. It's raining in Delhi, so I hope it's raining everywhere else too. Take care, and we'll see you next time.